Biology relies heavily on reading genetic code. We sequence a viral genome and treat that DNA as a master blueprint, assuming it tells us exactly how the final organism is built. But a genome is merely a set of instructions. Predicting the physical architecture of a mature virion exclusively from in silico DNA analysis often leaves us with an incomplete or even inaccurate model. To actually understand the mechanics of infection, how a virus targets, binds, and breaches a host, we have to identify the physical materials it uses. We have to map its final structural proteins. This presents a severe analytical challenge. A complete virion is constructed from proteins that vary drastically in abundance. Some form massive outer shells, while others are tiny, single-copy components of the injection machinery, making them incredibly difficult to isolate together. We cannot just sequence a virus to know its capabilities. We must physically dismantle it and map the pieces using mass spectrometry. This brings us to WKMV, a highly lytic bacteriophage that infects the bacterium Pseudomonas aeruginosa. Based on raw genomic similarities, researchers initially classified WKMV within the well-studied T7 supergroup of phages. In 2006, Dr. Rob Levine, Dr. Jean-Paul Nobin, and colleagues set out to examine that classification. Their study pursued two distinct objectives. First, they deployed two different mass spectrometry workflows to physically map the WKMV virion. Second, they used that structural map to test its assumed relationship to T7. By testing theoretical in silico predictions against hard proteomic evidence, this research possessed the potential to revise how we categorize viral taxonomy. The team's first analytical approach relied on fractionating the structural proteins using denaturing gel electrophoresis, commonly known as SDS Phage. Extracting clean data required rigorous sample preparation. The researcher used a sequential cesium chloride gradient centrifugation to separate the intact viral particles from host cell membrane contaminants. Looking at the gel analysis from this process, the first two columns illustrate the progressive disappearance of P. aeruginosa contaminants. The remaining visible protein bands, now mostly purified phage structures, are excised directly from the gel and digested for liquid chromatography mass spectrometry. The thick, dark bands in the third column yield high sequence coverage for abundant proteins. This run identified the dominant GP32 capsid protein at 38 kilodaltons and the massive GP36 internal core protein around 94 kilodaltons. SDS Phage provides deep, high-resolution data for dominant architectural structures. However, because it relies on extracting material from visually distinct gel bands, it remains completely blind to vital, low-abundance structural components. Finding those hidden minor proteins required a second, complementary workflow, whole phage shotgun analysis, or WSA. WSA bypasses the visual gel bottleneck entirely. Researchers dissolved whole virus particles directly, then digested the entire mixture with trypsin at once, creating a massive, complex peptide soup. To read this dense mixture, peptides undergo reversed phase HPLC, followed by mass fractionation inside the mass spectrometer. This method comes with a clear trade-off. WSA provides much lower overall sequence coverage for individual proteins compared to the direct gel extraction method, yet WSA succeeded exactly where the gel failed. It detected invisible minor polypeptides, specifically the GP33 protein, the GP47 protein, and the GP48 protein. Mapping a complete virion requires both pathways. Top-down gel fractionation establishes the major architecture, while whole phage shotgun analysis sweeps for hidden, low-abundance anomalies. With the mass spectrometry complete, the final methodological step takes the peptide data from both workflows and maps it back onto the theoretical open reading frames derived from the DNA sequence. This graphic shows the comparative phase of the study. The newly validated WKMV structural map aligns against the classical T7 genomic model. The mapped data immediately highlights a major structural deviation regarding the expression of the major capsid protein. The classical T7 model relies on a complex translational mechanism called ribosomal frameshifting to produce its major and minor capsid proteins during gene expression. The WKMV proteomic data, however, confirms a complete absence of ribosomal slippage in its major capsid sequence. This lack of slippage directly implies morphological differences in the phage head. It proves that WKMV operates under distinct functional requirements compared to the T7 standard. Moving down from the capsid head, the internal virion core and its mechanism of infection display an even starker contrast. In the classic T7 mechanism, the virus utilizes an N-terminal transglycosylase and an essential degradable plug, GP13, to keep the DNA channel closed. The WKMV map reveals an entirely different setup, lacking any equivalent to the GP13 plug. Instead, Levine's team identified a specific domain reorganization. WKMV relocates its functional lysosome domain to the C-terminal end of protein GP36. This C-terminal lysosome possesses highly effective peptidoglycan-degrading enzymatic activity, alongside strong thermostable properties. The mass spec data also revealed smaller structural proteins, GP38 and GP40, hinting at a unique heteropolymeric tail fiber structure, responsible for initial host adsorption. This structural domain reorganization directly dictates a distinct DNA ejection and host penetration strategy for the WKMV bacteriophage. The core objective of this study was to transform raw, complex viral lysate into a precise, validated physical model of a biological machine. In doing so, it uncovered three major physical deviations: the lack of capsid frame shifting, a reorganized injection needle without a degradable plug, and unique heteropolymeric tail fibers. These hard physical deviations force a reevaluation of established phylogenetic relationships based merely on DNA similarities. Because of these structural realities, WKMV, alongside its relative SP6, must be formally classified into a completely distinct subgroup, separate from classical T7. Genomic sequencing provides a necessary blueprint, telling researchers what a virus could theoretically build, but only rigorous dual-track mass spectrometry can prove exactly what it did build.